Today's lecture is the continuation of the previous lecture, and we are going to conclude classification of fungi in this lecture. We're going to look at the characteristics, significance, and life cycle of ascomycetes, basidiomycetes, and microsporidia. The first one is ascomycota. The members of this family are informally known as ascomycetes. They are named after their characteristic reproductive structure, which is like a sac. This is known as ascus. This one is the fruiting body of this fungi, which looks like a sac, and after that it is named as ascomycota. This sac-like structure is actually the reproductive structure of this fungi. Ascomycetes are ecologically important. They are living in freshwater, marine, and terrestrial habitats, and they are responsible to degrade many chemically stable organic compounds such as lignin, cellulose, and collagen. And many species, it's a very diverse class, and many species are economically important and well known. Some important ascomycetes includes red, brown, and blue-green molds that cause food spoilage, the powdery mildews that attack plant leaves, and the fungi that causes chestnut blight and Dutch elm disease. These are the diseases of the higher plants, vascular plants, and they causes the um, plant to die, their leaves wilt off, and ultimately they cause infection. Many of the yeast are also included in this class. There are edible morels and truffles also. The pink bread mold, Neurospora crassa, is also an important research tool in genetics and biochemistry, and it's also an ascomycete. Here, this is the powdery mildew, which is the infection of the plant. It looks like this, and this is the pink bread mold, which is known as Neurospora crassa. This is tuber brumale, which is highly prized for its taste by gourmet cooks, and this is Morcella esculenta, which is a morel, and it is the choice of choicest edible fungi that fruits in spring. A newer class of ascomycete, which is known as Archaeorhizomyces, was described in 2011. This new group is globally distributed, and it is found in association with the roots of pine tree, but it is not considered as a mycorrhiza because it can be grown in pure culture in lab. These are filamentous like Aspergillus and Neurospora, which are also ascomycetes, and they produce slender hyphae. Other ascomycetes, the significant species includes yeast, and they many of the ascomycete are showing life cycle that alternate between yeast and filamentous form. That is, they can produce hyphae as well, or they can reproduce like yeast by budding off from the parent cell. The most useful, the most valuable organism which is an ascomycete is Saccharomyces cerevisiae. It is an important research tool. It is used to study and understand cell division and loss of cell control that occurs in cancerous cell. Aspergillus species of this class is used to prepare citric acid, soy sauce, vinegar, and penicillium to produce the antibiotic penicillin. Some of the ascomycetes, such as Claviceps purpura and Aspergillus flavus, are pathogenic to higher plants. Claviceps purpura in particular, it parasitizes rye and other grasses, causing the plant to for causing the plant a disease known as ergot. It can be seen here like this. This is a rice plant. This is the grain And in this grain, this black structure, a stem-like growth, a hardening, is known as ergot. This type of um, infectious grain, if it is eaten by any animal or human, then it can cause a condition which is known as ergotism. Ergotism is accompanied by gangrene, psychotic delusions, nervous spasms, abortion, and convulsions. It is the cause of major outbreaks in history in France and England in Middle Ages, and it is also known to be associated with witchcraft that occurs in the 17th century. Uh, Ergotism is the conditions which is a kind of a fatal condition. It is actually due to the active ingredient that is present in this fungi, which is LSD, lysergic acid diethylamide. It is also used as a drug abuse. Uh, Apart from this, several Aspergillus species are also noteworthy, like Aspergillus fumigatus, which is ubiquitous in the environment and is commonly found in homes and workplaces, etc. Aspergillus nitulans, which is a model organism and it is used to study the questions of eukaryotic cells in developmental biology, and Aspergillus oryza, which is used in the production of traditional fermented foods and beverages in Japan, including sake and soy sauce. Most of the fungal species that infect animals including humans are ascomycetes, and many are opportunistic pathogens as well. The most common in this genera is Candida, Blastomyces, and Histoplasma, which causes different diseases in humans. And in addition to this, there is another ascomycete which is known as Stachybotrys chartrum, and it is the cause of sick building syndrome. And this picture, actually these are the conidiophores which are it's multiplied several times to show this is the conidiophore, and it is the cause of this sick building syndrome. Actually, it's also a very common fungi. It can grow anywhere, and it grows especially in those areas which are not well ventilated. Aspergillus toxin, known as aflatoxins, are an important cause of food contamination, and this kind of toxin can result in liver cancer as it ruptures liver cells. Most of the ascomycete, they reproduce asexually through conidia, which I have shown you in earlier lectures how this conidia is produced in conidiophore, and these are the major form of dissemination, that is distribution. But some filamentous fungi, they also produce sclerotia. Sclerotia are the compact masses of hyphae, a dense mass that can survive the winter, and it is very advantageous for the fungi as it remains viable in the soil. And when the temperature rises, it gets its nutrients from the soil, and then it starts to reproduce by making more conidia or hypha. Now, if we talk about the life cycle of ascomycetes, it's quite simple. As you can see here, that the asexual reproduction is quite straightforward and simple, as the mycelia will grow conidiophore, and in this, by cell division, it will produce conidia. These spores will again germinate into a new mycelia. Now, if we talk about the sexual reproduction, it involves the fusion of two different strains, one male and one female. The male strain or male hypha is known as antheridium, and the female hypha is known as escogonium. When these two mycelia come close together in plasmogamy, they undergo mitosis. They fuse, but they do not fuse together. They actually form a new mycelia, which is known as ascocarp. In this ascocarp, the tip contains asci, in which the in this asci, one cell from the ascogonium and one cell from the antheridium are present here. Now, what happens in karyogamy? These nuclei, which are separate in this asci, ascus, they fuse together to form a diploid zygote. And now this zygote will undergo meiosis and form 4 haploid nuclei like this. Then it will go mitosis, undergo cell division, and these 8 haploid ascospores are formed, which have distinct walls, and they are placed together in one ascocarp. And then they again disperse and germinate into another mycelia, and in this way this life cycle continues. So in plasmogamy and mitosis, the two mating strains, they come closer, and then they form another mycelia, which is a specialized mycelia known as ascocarp, and that contains ascus, the fruiting body. Then it will undergo karyogamy, and then it will produce the zygote. Now this is for your convenience, so you can get a summary of this whole life cycle of ascomycetes. Now we move on to the other class. Now first of all, by moving on further, I want you to ask yourself this question and think about it, go through the previous slide, read it, and then answer this question. Which of the following statements is true? A dikaryotic ascus that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores, or a diploid ascus that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores. A haploid zygote that forms in the ascocarp undergoes karyogamy, meiosis, and mitosis to form 8 ascospores, or a dikaryotic ascus that forms in the ascocarp undergoes plasmogamy, meiosis, and mitosis to form 8 ascospores. You have to test yourself, answer this question, and I'll discuss it in our live class or live session. The next class is Basidiomycota. These are also known as club fungi, and most commonly found mushrooms that we eat are included in this class. So mushrooms, puffballs, shell fungi, they all are members of this group, and they are also the cause of very famous plant diseases, rust and smuts. These groups contain almost 15,000 known species, and it is distinguished by the presence of a club-shaped reproductive organ, which is known as basidium. Basidiomycetes are named for their characteristic structure or cell, the basidium, which is involved in sexual reproduction. It is formed at the tip of the hypha, and it is normally club-shaped. 2 or more basidiospores are produced by this basidium, and these basidia are held underneath a fruiting body, which is known as basidiocarps. Basidiocarps are like, you can resemble it with the ascocarp. It's just like that, but it's a club shaped. It's more you can say in width it is more circular and elongated, and it can reach almost 60 centimeter in diameter, and it has many spores, and it is lined with basidia that produces basidiospores. A single fruiting body can produce millions of spores because it is lined with many of the basidia. Mushrooms, which are used in food, and the cultivation of mushroom Agaricus campestris is a multi-million-dollar business, and this mushroom is included in this class. They are also saprophytes. Mostly they are saprophytes. They decompose plant debris, especially cellulose and lignin, and the common fungus Polyporus squamous is also found in dead trees. Some common pathogens of this class are known as uridinomycetes and stellagminomycetes, and they cause the most famous rust and smut disease. Rust and smut disease are economically they are a very considered important diseases because they destruct cash crops like corn, wheat, etc. So, in the rust disease is due to this fungi basidiomycetes, and a slight difference between rust and smut is that rust disease requires almost two hosts to complete its life cycle, and it is usually localized to a certain area of the plant. For example, if it is infecting the stem, then it is localized over there, whereas the smut appears in whole plant and it completes its life cycle in a single host, and that is always a flowering plant. So a stellagminomycete, stellagomyces, is a common corn pathogen, and it has become a model organism for studying plant smuts. Many mushrooms produces alkaloid that act as poison, or they can cause hallucinogens. One such example is death angel mushroom, Amanita phalloides. Amanita phalloides actually uh, two types of alkaloids are being isolated from Amanita phalloids. The first one is phalloidin, and the second one is alpha-manidin. Phalloidin attacks the liver cell, it ruptures cell membrane of the liver cells, and alpha-manidin attacks cell lining of the stomach cells and causes GI symptoms that are associated with mushroom poisoning. Many Amanita muscaria is also poisonous and hallucinogenic. So, the Basidiomycete Cryptococcus neoformans is also an important human and animal pathogen, and it causes disease known as cryptococcosis, which is a systemic infection, and it involves primarily the infection of lungs and central nervous system. Now, if we look at the life cycle of Basidiomycete, it is also quite simple, but it is this stage, the dikaryotic stage, is actually the dominant one. The spores are actually uh, formed from the reproductive organs, the basidia. Now, these basidia are underneath these basidiocarp. This is the club-shaped basidiocarp, and it contains many types of basidia, which forms two basidiospores. Now, what happens in this basidia? It undergoes karyogamy, and it contains diploid nuclei to form a zygote that contains uh, cells from both strains. So it's a diploid zygote. It will undergo meiosis and forms four haploid nuclei in the basidium, and these will go and further it will go cell division, undergo cell division, and basidiospores are formed. These are these four nuclei are included in one basidium, and then a cell division will occur, and it will produce four basidiospores, and then they will disperse and germinate, forming these haploid cells to form new mycelia. Now, when these undergo germination, then they have different types of mycelia. For mating one of the positive side and one from the negative side, these two types of strain uh, come closer, they mate together in plasmogamy, and this fusion between these two different mycelia which are compatible, that results in the formation of dikaryotic mycelium. Now, this one is the dominant phase of this life cycle. This dikaryotic mycelium, as it contains it develops. This one is the primary hypha, And this one, when this primary hypha fuse together in plasmogamy, they give rise to this secondary mycelia, this one, and it contains one cell from this strain and one cell from this strain. This specialized secondary mycelia will then undergo mitosis in right environmental conditions, and then they will form basidiocarp or gills which are having basidia, and then it will undergo karyogamy where they will form zygote, and this cycle will continue like this. Now for your convenience, I have explained it in these steps so you can see there. The next and the last class of this classification towards microsporidia. Of all the fungi, this member of microsporidia are the most confused in taxonomic history, as these microorganisms are still unsettled, and many scientists consider them protist. More than 1200 species are being catalogued, out of which 143 are placed in different genera, and 14 species, at least 14 species, are human pathogens, which are very important human pathogens. They are actually obligately endoparasitic microbes, which means that they require certain type, specific type of host to complete their life cycle. So still some consider them protists, but however, molecular analysis of the ribosomal RNA had a study of specific proteins has revealed that they are actually a sister branch of fungi, so they are being placed under fungi. Unlike other fungi, they don't have mitochondria, uh, peroxisomes, and centrioles, but they contain mitosomes. Microsporidial morphology is also unique among eukaryotes because it contains a distinct type of an organelle which is known as polar tube, and this polar tube actually helps this um, fungi to grip on the host cell, and it gets an entry from this microtube, polar tube. Thus, they are actually viable spores of almost 1 to 4 micrometer, which are viable outside the host cell. And you can see here this one. This is the spore. It's almost oblong type of a spore, and as you can see here that it lacks mitochondria and other specialized organelles. However, it contains nucleus, and this is the polar tube. It's very specific, very distinct, and from this we can easily identify that any infection is caused by this microsporidia. They lack the certain organelles, but their cell wall contains chitin like other fungi. So the spores may be of different shape, spherical, rod-like, crescent shape, or like egg, and especially they are problematic for those people who are immunocompromised, such as in HIV or AIDS. So they cause infections in immunosuppressed or immunocompromised patients. This polar filament, when it wants to anchor itself to the host cell, it jacks its polar tube with a force such that this anchoring disc will go and stick itself to the host cell membrane. From there it will pass on its protoplast or polar plast in this case to the host cell, and then it starts to form its life cycle by making more spores inside the host cell and then taking control of that host cell and causing infection. Now let's take a quick recap. Ascomycota are the members of this class are known as sac fungi because they contains the reproductive structure which is sac shaped, called ascus, and They have either a yeast morphology or a mold morphology. Mold morphology means they have filamentous producing hypha, or yeast morphology means they are unicellular, they can reproduce themselves by budding. The important ascomycetes include the yeast Saccharomyces cerevisiae, which is very important in baking and brewing industries. Aspergillus species are also very important. Claviceps purpura, which is the cause of ergotism, and the indoor mold is Stachybotrys tortrem. Basidiomycota, the members of this class are known as club fungi. They are named after their basidium that produces basidiospores, and genera from Uridinomycetes and Stalaginomycetes. They include important plant pathogens that causes rust and smuts. Then comes Microsporidia. The members of this class have a unique morphology because they contains polar tube. Some of the scientists consider them as protists. They include virulent human pathogens, and some can infect other vertebrates and insects as well. That's all for now. We'll just continue our lecture in the next class. Thank you.